Now we are going to talk about the various tools which are involved in the process of rDNA technology. Now what is recombinant DNA technology all about? I am going to summarize it in 3 steps so that you get an idea what each tool is going to be useful for. We must know what the process is all about. A brief introduction was given to you in the previous lesson. Now we are going to see the 3 steps in which this entire process is carried out, and from that you have to understand what tools are being used for that process. Okay. So the first step is identifying the gene of interest. This is the first step in obtaining a genetically modified organism that could be present in any living cell. From there you have to identify the gene of interest. Then that gene of interest has to be cut and transported to a suitable vector. Okay. So we have to make sure that incorporation of gene of interest into vector DNA, and the third step is introduction of vector into suitable host cell. Suitable host for multiplication. Okay. So we have the gene of interest. We have that gene of interest being put into a vector DNA, what that vector would be we will see, and lastly that vector DNA along with the vector has to go into a host for multiplication, which is known as gene cloning. Now coming back to the topic of this lesson, that is tools. What all tools will be required for carrying out these 3 processes or these 3 steps which result, all these 3 when combined they give us a genetically engineered or genetically modified organism, that is known as GMO. It could be a genetically modified crop, it could be a genetically modified animal, it could be a genetically modified bacteria, whatever we are talking about, that would be genetically modified because the identified gene of interest would be alien to the host DNA or to the vector DNA. So we are having a recombinant DNA. What was recombinant DNA? DNA which has some extra DNA in it, which was not originally the composition of that DNA. So we have the genetically modified organism. For carrying out these 3 steps, we need certain tools, and what those tools are, we are going to see that. First of all, the gene of interest, when it has been identified, it needs to be cut out from there. So we have the first tool, that is Restrictionenzymes. As I told you in the introductory lesson also, that these are a special class of nucleases. Nucleases are the enzymes which are responsible for cutting the DNA. So the gene of interest is nothing else but DNA sequence only. That DNA sequence has to be cut from there and brought in contact with the vector. So the second tool is vector, which will incorporate the gene of interest in itself. Okay, just join that gene of interest that has been cut from the organism, join that gene of interest into it, and then as the vector goes and infects an organism, it is going to infect the particular cell with a high dividing ability. Okay, and that particular cell would be a suitable host cell that is ready to take up the recombinant DNA which is present inside the vector. So these are the 3 tools which are required for carrying out the recombinant DNA technology in order to obtain a genetically modified organism, which is obtained by following these 3 steps. Now, these tools are integral to these 3 steps because first of all you have to cut the gene of interest, that would be cut by restriction endonuclease. Then you have to put that gene of interest into such a DNA which has the ability to go somewhere and produce its copies in that given environment. That such DNA that is used, that is nothing but vector, and that such DNA has to go in an environment where it can produce its copies. That environment would be provided by a suitable host cell. So, these are the 3 tools that you must have in at your dispense, so that you can carry out these 3 activities and obtain the result of a genetically modified organism, which is the prerequisite for carrying out this recombinant DNA technology. So, this is all about the tools which are required for recombinant DNA technology. One by one we are going to dedicate a lesson to each of these and understand what is their purpose in this entire process.